The building blocks of life and fluid balance. When we're dealing with people, we have to be able to recognize what is abnormal. If you cannot recognize abnormal, then you're going to have a problem treating your patients. And in order to understand what is abnormal, you have to understand what is normal so that you know when it's not normal. So the structures of the body are designed to carry out all of their functions very precisely, and it's really kind of amazing how well they actually do this under normal circumstances. When we're talking anatomy, we're talking about the structures themselves, and then physiology is the function or actually carrying out of that function. Our cell is going to be our basic building block of life. It contains cytoplasm and your organelles. Groups of these cells form our tissues, which then form organs, and then eventually form body systems, which form us. So our cells are composed of chemicals that are surrounded by a chemical environment. We know chemistry is the study of composition of these substances and how they interact and work together. Because of this chemistry, because of the way they are designed, they actually allow the cells to engage in a process called metabolism and maintain homeostasis, which homeostasis is just the overall inner balance inside the body despite your environmental changes. So it's keeping it balanced and level. Now, a chemical element is something that can't be broken down any further, and our atoms are our simplest unit of an element. Now, we do have subatomic particles. These are going to be electrons, which hold a negative charge, protons, which hold a positive, and then neutrons, which are neutral. This electrical current that flows from an area of positive charge to an area of negative charge, and the attraction of these oppositely charged particles is what kind of allows these substances to combine and change structure. Our atoms are capable of losing or gaining electrons. So an ion is just a group of atoms or an atom that can carry a positive or a negative charge. A positively charged ion is called a cation. I always remember this one because it's got a T in the middle of it. It looks kind of like a plus sign, positive. And then anion is a negative charge. An anion, in medical terminology, an or a at the beginning of something means without. So this is without a charge. Now, an electrolyte is a substance that results when chemical compounds dissociate or break down into negatively and positively charged ions, these anions and cations. Example, like table salt. So salt is sodium chloride, which is represented by sodium as Na and chloride as Cl. So when it is dissolved in water, it dissociates into your positive sodium cations and your chloride anions. You have quite a few chemical elements within the body, but you have 4 primary chemical elements that compose most of your body's atoms, and these are going to be hydrogen, oxygen, carbon, and nitrogen. As we know, water makes up about 60% of our overall body composition, and all of this water is contained in 2 major compartments within the body. So these are going to be your intracellular compartment and your extracellular compartment. So your intracellular fluid is the total amount of water within the cells, and this is the greatest percentage of water, about 75% of that water within the body. Your extracellular fluid is the fluid that's outside of the cells. It's further divided up into intravascular and interstitial. So intravascular is within the blood vessels themselves, and interstitial is within the tissues themselves. When we're dealing with solutes, these are just formed particles in your bodily fluids. So it could be electrolytes, carbohydrates, proteins, lipids, drugs, all these different things. The concentration of these solutes in these fluid compartments influences the movement of the water in and out. So the movement of water along this gradient from lower concentrations to higher solute concentrations is called osmosis. And your body's always going to try to equal out the water and solute movement. So there's a continuous movement of all of these things in your body. Now, the difference in the concentration of your solute creates what's called an osmotic pressure. And so this is basically the ability to pull water across the cell membrane from a less concentrated solution to a higher concentration to try to equalize out the two. Now, oncotic pressure is a portion of the total osmotic pressure, And this is created by the concentration of large proteins that can't pass easily through the capillary wall. The capillaries are very permeable to small ions, so the oncotic pressure is equivalent to the total osmotic pressure in these vessels. Oncotic pressure returns fluid to the bloodstream when it's forced out through the capillary walls under the hydrostatic pressure created by the force of the blood. So this higher concentration of albumin in the bloodstream kind of pulls the water back into the capillaries, which prevents edema. Your osmotic pressure of IV fluids is called tonicity. This tonicity is measuring essentially the milliosmoles per liter of fluid. So when a fluid is equal to the bodily fluids in the amount of its solutes, it's said to be isotonic. The osmolarity of the body is between 280 and 310 milliosmoles per liter. So if we're talking electrolytes, your sodium chloride, 0.9% sodium chloride, which is also called normal saline, has a concentration of 310 milliosmoles. So that's considered to be isotonic, which means when it's given, this isotonic is not going to create an immediate movement of water between the extravascular and the intravascular compartments. Now, a more concentrated solution that's higher in solutes is considered to be hypertonic. And so this is going to have a higher osmotic pressure, usually greater than 350 milliosmoles, that's going to move water, cause it to move from within the red blood cells to the intravascular space, which can actually cause destruction of the red blood cells. An example of this hypertonic IV fluid would be 2% sodium chloride. A less concentrated solution, such as 0.45% sodium chloride, is considered to be hypotonic, so it has a lower osmotic pressure, usually less than 250 milliosmoles. When you have a hypotonic solution added to the bloodstream, the water is going to leave the bloodstream and enter the interstitial fluids and cells. And as a general rule, if you want to remember how the concentration of the solutes affect water movement, wherever sodium goes, water follows. But keep in mind that's only with osmosis. The movement of your sodium and water can also be affected at the cellular level by your active transport mechanisms. One of these active transport mechanisms is your sodium-potassium pump, and it's a protein on the cell membranes that keeps ions at proper levels inside and outside of the cells. Your principal intracellular cation is going to be potassium, and your principal extracellular cation is going to be sodium. So they're both located on both sides of the cell membrane, and sodium is able to enter the cell from the extracellular fluid, but it can't leave the cell because its concentration is greater on the outside of the cell. Problem with that is if sodium doesn't leave the cell, water is going to keep coming in because it's going to follow the salt, and it's going to come in quantities that's going to make the cell burst, which causes lysis, L-Y-S-I-S, which is cellular death. The potassium pump is going to move 3 sodiums out of the cell for every 2 potassium moves into the cell. So this difference in the concentration of these ions creates this negative electrical charge inside the cell that creates a membrane potential. The sodium-potassium pump requires energy to move this sodium, so it's going to require ATP, which we will discuss further.